Hello, I'm Eric Sorscher, the Doug Hertz Endowed Professor of Cystic Fibrosis Research at Emory. Doug is a humanitarian and a respected champion for many diseases of children, and cystic fibrosis is high on that list. I began my work on cystic fibrosis as a medical student in Boston. I spent a lot of time at Children's Hospital. This was 3 decades ago. Cystic fibrosis was a very different disease then. I was playing guitar on the ward of Children's Hospital that took care of patients with CF, and I met young kids with respiratory failure who were dying, and also young adults. And it felt so important to me that I left medical school, and I've never really looked back. My passion has been working on CF ever since those days. Our laboratory works on finding new and effective therapies for everyone with cystic fibrosis. For many years, I co-chaired an international consortium with members from the UK, other parts of Europe, Canada, and the United States. And one of our main jobs was helping industrial partners identify better compounds to treat people with cystic fibrosis. Over those years, we watched as CF changed from a disease that was virtually untreatable in terms of its basic molecular problem to now a very treatable condition. And in that time, I've become chair of the National Medical Advisory Council for the Cystic Fibrosis, and also a trustee on the National Board of Directors for the CF Foundation. Now, what's really changed things in cystic fibrosis has been a technology called high-throughput compound library screening. And it's really turned the world of drug discovery on its head. In the past, the way you developed a drug for a disease like CF was to learn everything you could about the molecular biology, the genetics, the physiology, the pathophysiology, the cellular biology, and then use that knowledge to do your best to come up with something that would help. High-throughput compound library screening turns that on its head by taking a library of 100,000 or 500,000 or even 1 million compounds and giving that library to a robot and asking the robot to screen every one of those drugs against cells that have CF. And that way hits are found, drugs that can help people with the disease. And through that technology now there are drugs for upwards of 90% of people with cystic fibrosis that treat the basic molecular defect. Not all of the 90% of people with CF who have a drug will benefit from it, but many will. And for those that benefit, it can be really remarkable. Within 4 weeks, patients feel much better. They can breathe better than they have in years. Lung function improves objectively and symptomatically, and it's really remarkable. It's been a remarkable thing. It's like the answer to a dream that many of us have worked on for decades to help people with this disease. Now, though 90% of people have a drug, that means 10% are at risk of being left behind. These are often people with rare variants, and it's a terribly difficult situation. And our laboratory is devoted to find ways to treat these patients. Many of them have rare forms of the disease, as I said. So often the major pharmaceutical companies are less interested from a strictly financial standpoint in drug discovery for the last 10%. Our group has performed 600,000 assays using robotics and high-throughput screening with high-end food and well-recognized, internationally recognized pharmacologist Hugh Davies, who's known around the globe for his work in chemistry. We've now discovered drugs that blunt lung inflammation and overcome problems associated with rare variants, especially those in the last 10%. There are ongoing issues with the new and emerging drugs for CF. They include cost and access among those who don't have a drug right now. You can imagine what it's like to be in the group of patients who are at risk of being left behind, those 10%. They've always been in the same boat. Everyone with cystic fibrosis has always had the same outlook. Now 90% have a much brighter and optimistic outlook on the disease, and 10% still with the same prognosis. So our laboratory focuses on that group, and we've come up with better ways to predict response to the new drugs so that more patients can get access to these therapies. We develop new markers that demonstrate drug response and clinical improvement. We develop a better understanding of things that can go wrong with the CF gene and how to overcome these problems. And we really survey every gene in our DNA to find new molecular targets that will help people with the disease. All of this with one goal, the goal being to help people with CF. It's an amazing story of a common goal to achieve something good, helping people with a serious disease. And the story is emblematic of ways that working together towards an important objective can be successful. It's a story of what we can do if we devote ourselves, patients, families, friends, communities, fundraisers, caregivers, and scientists. When I was a boy, death in childhood was very common from CF. Now it's very common for people to live into their 40s and well beyond. And it's our hope that now 90% of cases will have substantial prolongation of their lives and of the quality of their lives. And there are new technologies that may help things improve even further and address the cost of these drugs. Gene editing, gene transfer, better anti-inflammatory molecules, better antibiotics, all can contribute. With your support, with your help, we can make a better life possible for everyone with cystic fibrosis. Philanthropy can make this possible. In our own laboratory, we need equipment, recruitment funds, and other resources to facilitate testing of new therapies for CF for people with the disease. And we thank you so much for listening and for considering our work, and wish you the very best. Thank you.